Hello everyone, welcome back to my channel. I hope you all are doing well and you are also preparing pretty well for the upcoming examination like CSIR. So you already know that I provide so many important video related to different competitive examination like CSIR, DBT, GATE and other biology related exam. So you can check all my videos because all are free of cost and every video is in detail, and you can also practice different questions from my video. So before moving forward, don't forget to subscribe the channel and motivates me a lot, and do watch the video till the end. So you know that in every exam like CSIR, DBT, you can say even GATE, the questions are directly from life science standard book like Cooper, Karp and Lehninger biochemistry book, and whatever be the books recommended for life sciences. So basically all the questions are framed directly from the standard book content. So if you don't know how to read that book and how to note down important questions from it or important pointers out of it, so this video is for you. From this video you will be able to recall that what can be asked in your exam. Compiles different questions from the Cooper book, that is for the cell biology. So you can check it out that how the questions are framed. So a lot of efforts was required to make that. So don't forget to subscribe the channel for more such videos. So this is the first question. So the first question is, which of the following statement is true regarding genome size and biological complexity? So you have to relate that is there any relationship between the genomic size of the organism and the biological complexity. It means its genomic material. It has to do with its genome size or not. So this is purely extracted from whatever written in Cooper. So genome size always correlates with biological complexity. Larger genome always contain more protein coding gene. Organisms with larger genome are always more complex than those with smaller genomes. Genome size does not always correlate with genetic or biological complexity. Yes, this is the right answer, because it is not at all necessary that if an organism is small, its genome will be of small size. No, it is not at all related. And also if the organism is large in size, it is not at all necessary that its genome size will also be huge. So there is sometimes there is a correlation and sometimes it is not. So that is why all these three options are wrong, because genome size not always correlates with biological complexity. And also it doesn't mean that larger genomes contain more protein coding gene. This is not at all true, because it is not necessary that whenever an organism is having a larger genome, it has all the coding genes, because we have a larger proportion of non-coding genes also. So this is also wrong. So the right answer for this question is D. The passage clearly states that genome size is not always related to genetic or biological complexity. Now next question. Which organism has a genome size over 10 times larger than that of humans despite being less complex? So it is the salamander, as per the passage or the paragraph written in Cooper. So किस organism का genome size 10 times बड़ा है human से, and उसके बाद भी वो complex नहीं है? So it is the salamander. Salamander and lilies are mentioned as examples of organism with genome size 10 times that of humans. So you can remember lilies and salamanders have 10 times larger genome than that of the humans. So next question. The number of genes in human genome is approximately. So this is a factual question. In CSIRB, you can expect this question. So in humans, we are having about 20,000 genes in our genome. So 20,000 protein coding genes are there. So the right answer is option B. Now next question. Which of the following organism has a genome only about 5% of the size of human genome yet has more coding genes? So it is none other than the Arabidopsis thaliana, the model organism which people used to study a complex mechanism in the Arabidopsis thaliana. So Arabidopsis thaliana has about 26,000 protein coding genes, more than the humans. and it has a smaller genome size. So the first question, that the smaller size doesn't mean that the organism will have a lower number of genes or a decreased number of genes. No, it is not at all. So Arabidopsis thaliana has a smaller genome size, yet it has more protein coding genes than humans. So now next question. How many times more genes does the human genome contain compared to E. coli? So basically these are all comparison, that humans have how many genes, bacteria E. coli have how many genes. So as per the paragraph or the content taken from the Cooper, you can say 5 times more. Like, how many more genes does the human genome contain compared to E. coli? E. coli is a bacteria, prokaryote. Humans are eukaryote. So roughly 5 times higher genes we have in our genome as compared to the prokaryotes. So human genome is 1,000 times larger. It contains only about 5 times more genes than the E. coli. Now next question. The paradox where genome size does not correlate with organism complexity is known as C-value paradox. So this is a term when there is a lack of correlation in value paradox. So now moving on to the next question. So you can see this diagram, the genomic size, the range of sizes of the genomes of representative group of organism as shown in the logarithmic scale. So you can see how many base pairs per haploid genome are there in different group of organisms. So you can see the bacteria, the yeast, fungi, plants, insect, amphibian, birds and mammals. So there are a lot of variety in between the genes of organisms. So you can see the plants like Arabidopsis and lily have higher genes than as compared to the humans. So and also you can see salamander has a higher gene than the humans. So through this diagram you can compare the genomic size of each and every organism. So this is an important part. Questions are asked from this topic very oftenly, so don't forget to include this in your notes. Now moving on to the next. Now another question, what is the primary reason for the larger genome size of many eukaryotic organisms? So what is the reason that eukaryotes have larger genome size? So high number of protein coding genes? No, we just now read that it doesn't mean that if the genome size is higher or larger it has more protein coding genes. No. Repetitive translation of mRNA? No. So if it is repetitive translation, it means so many proteins will be synthesized. So this is also wrong. Large amount of non-coding DNA. Yes, this is the right answer. Multiple copies of mitochondria? No. So the variation in eukaryotic genome size is mainly due to differences in the non-coding DNA, not the number of protein coding gene. As I told, we have majority of genome size is full of non-coding DNA. So this is the reason of our large genome size. Now next question. Which of the following correctly defines an intron? A segment of DNA coding for protein, a DNA sequence that regulates replication origin, a non-coding sequence within a gene removed during RNA splicing, a bacterial operon controlling gene expression. So as we all know, an intron is a non-coding sequence within a gene removed during the RNA splicing wala part. We remove the intron so that the non-coding sequence are removed, and this process is known as RNA splicing. So introns are the non-coding region that interrupts coding, yani introns, exons like this. So this is the right answer. Now quickly moving on to the next one. Exons in eukaryotic gene refer to opposite of intron. They are the protein coding regions retained in the mature mRNA. From the pre-mRNA, we eliminate the intron, that is the non-coding one, and we only keep the protein coding regions, that are the exons. So right answer for this question is option B. Now moving on to the next question. The discovery of introns in adenovirus was first made by observing out of the following what? Northern blot, SDS page, RNA DNA hybrid, southern blotting of viral genome. So it is obviously the RNA DNA hybrid formation under an electron microscope. So this is again a very important question, a factual question which can be directly asked in your CSIR exam. So we always forget to make pointers out of whatever we are studying from the standard author book. So this is not a good way to do so. This is not a good way to approach any exam. So try to watch every part which I will provide to all of you. I will extract questions from different standard books so that it will be easy for all of you people to include all this important point in your notes. So only you have to like, share and subscribe the channel. So now next question. RNA splicing results in what? Just now we have read that it is for removing the non-coding RNA and to join the exons which are the coding genes. So removal of introns and joining of exons in the pre-mRNA. So this is known as the RNA splicing. So RNA splicing removes the non-coding sequence, that is intron, and it joins the exons to form mature mRNA, which will be ready for the translation, that is protein synthesis. Now next part. Which virus helped uncover the phenomena of RNA splicing? Through which of the following phenomena we have unraveled the mechanism of, you can say, RNA splicing? So it is just because of the adenoviruses. Adenoviruses was used in early studies and they led to the discovery of RNA splicing. So please note it down in your notebook. All these questions are very, very, very important. Now next question. Who independently discovered the presence of introns in eukaryotic genes? So you have to tell the names of the researchers who contributed to the development of or the discovery of the presence of introns, that is a non-coding sequence in the eukaryotic gene. So it is Philip Sharp and Richard Roberts. So write it down in your notebook. Philip Sharp and Richard Roberts was the one who introduced the concept that eukaryotic genes also have non-coding sequences, like they have introns in their pre-mRNA. And also for this they received the Nobel Prize. Now next question. The genome of adenovirus is approximately how much? So it is about 3.5 base pairs, 3.5 into 10 to the power 4 base pairs. So you can write it down. Again, it is a factual question. So it is a suitable model for gene expression studies. So this is all for today. I hope you liked this video. So don't forget to subscribe the channel, because people watch and forget to subscribe. So only aim through this channel is to provide quality content and free of cost content. So you don't need to miss any video, or you don't need to pay any amount for it. So you only have to watch the video and write it down in your notebook. So if you like the video, don't forget to like the channel, and thank you so much for watching.